Evelyn Smith from Seattle U, rising sophomore working at the Allen Institute in the Lein lab.
Hello, everybody. You're halfway through, so congrats. My name's Evelyn, and I worked at the Lein lab at the Allen Institute, and my summer project was on variability in RNA quality across the human brain. So to give you a little bit of context about what the Allen Institute is doing as a whole, they're working on building these transcriptomic maps of the entire human brain. And they're essentially identifying each individual cell and what genes they express so that they can categorize them into cell subtypes and determine their functions. And the important thing about this research is that you need to ensure that the brain tissue is high quality. And the way that we do that is using something called an RNA integrity number. So what is an RNA integrity number? So an RNA integrity number, or a RIN score, is a metric that we can use on a scale of 1 to 10. So 1 being poor RNA quality, 10 being excellent RNA quality. And when you run a RIN test, you get these results. So you get a RIN score. This one has a score of 9, and you get a RIN graph here. And a couple important regions, I want to point out the 18S and the 28S fragment. And these are important because we calculate RIN score as a ratio of the area under these two peaks versus the area under the rest of the graph. So that's going to be important later. Maybe I'll quiz you on it. So why is this important? The Allen Institute typically only samples for RIN from the front and the back of the brain, but we weren't sure if that was representative of the entire brain. So here I am dissecting. That's a little sneak peek of the methods. But the main goal was to determine if RIN varies across regions of the brain. And then secondarily, I wanted to determine if RNA concentration impacted RIN. So if I take a slightly larger sample of brain that has a higher RNA concentration, is that going to skew my RIN score? We don't want that. I broke down the methods into 3 sections, starting with tissue dissection, arguably my favorite part. I actually really enjoy dissections. And if you imagine a brain and you split it into its 2 hemispheres, and then you slice the hemispheres from front to back, you'll get a brain slab like this one. So this is an example of a real human brain slab that I dissected. And primarily, we dissected from 8 anatomical locations. So dorsal, lateral, medial, ventral, etc. And that we did that to a total of 12 slabs. So we got 90 samples out of that. And the first donor, we did a front to back, pretty thorough sampling. And then the second donor was just for some additional data points so we could track trends. And then 5 of these samples were excluded for a reason I will explain on the next slide. During stage 2, which was RNA isolation. So the goal of RNA isolation is to isolate the RNA, as you could probably figure out yourself. But basically what we do is we mash the brain tissue up and we get rid of all the other cell parts so that we just have the RNA. And then we send this RNA to another lab at the Allen Institute, the RNA sequencing core, and they do the RIN testing. 5 of our samples did have to be excluded because the kit that we use for RIN testing requires a RNA concentration of 25 micrograms per microliter. So those 5 were below the threshold and we couldn't use them for data analysis. Just a side note, this is a NanoDrop, which is my new favorite machine that I learned how to use in the lab. Final stage was the RIN testing. So in the upper right corner here, you can see the bioanalyzer machine. And then this is the chip that we use. It's a gel electrophoresis system. So we take the chip and we load gel into it. And then we load the samples into these individual wells. If you can see that, it's pretty small. And then we load the chip into the bioanalyzer. And what happens is there's an individual electrode for each cell. And there's an electrical current that sorts the RNA fragments by length. And you end up with these lovely graphs here. So these are some graphs from the samples that I processed this summer. And just to point out, this one is 9.6, super high RIN score. And as you can tell, there's a really clear 18S and 28S fragments, like I pointed out earlier. But if you look at this one, 7.4, you see all that noise. So that's a sign of RNA degradation, potentially. 7.4 is still a good RIN score, but just so you can tell the difference. Now you know how to read RIN graphs. Moving on to results, the most important part. So we took those 85 data points and we did some statistical analysis using Python because I learned how to code this summer, really exciting. It's actually pretty fun. And we found this. So there's no significant difference between the distribution of RIN scores in the various anatomical locations. And you can tell that by visually through the plot. All the distributions pretty much overlap and they're in the range of 7 to 10, which are really strong RIN scores. And then we backed it statistically using an ANOVA test, which had a p-value of 0.7. If you took a stats class sometime at some point in your educational career, you know that this is insignificant. So we moved on from that. And then we checked slab number. And this one was actually kind of interesting. So we found a slight positive correlation between slab number and RIN score. So remember, slab number, we slice the brain front to back. 1 is going to be in the front, and then there's about 40 slabs roughly in your brain, depending on the size. So as slab number increased, you can look at this graph across both donors, they're color-coded. RIN score was also slightly increasing, and that was backed up with a p-value of 0.0006. So that one is significant. And just in case you might be thinking, maybe you were taking larger samples as you were going along, it could have to do with RNA concentration. Well, we just proved that using a RNA concentration versus RIN score scatter plot. And as you can see, there's no clear trend here, which is good news because that means if we take a slightly larger piece or a slightly smaller piece, it's not influencing our RIN. So to conclude, RIN sampling can continue as usual for future experiments. Basically, the correlations that we did find are not significant or not great enough to influence the protocol at this time, so RIN sampling can continue. Thank you. Special thanks to Dr. Ellenbogen and Dr. McDonald and everyone who makes this program possible. And then thank you to Erin in the back there for introducing me or welcoming me into your team at the Allen Institute this summer. It was lots of fun. Yeah, thank you.